So good evening, everybody, and thank you all for being with us tonight, the second night of Brain Health Awareness Week. And if you missed our first evening, that night was dedicated to ways we can keep our memory sharp throughout the lifespan. And you can still find that on our website at the link. So to introduce myself, my name is Ruth Slack, and I'm the director of the University of Ottawa Brain and Mind Research Institute. And before we start tonight's session, I'd like to introduce my Brain and Mind team who worked tirelessly to put on this entire week of events. So I'd like to introduce Natasha Hollywood, our program manager, Victoria Rache, our communication coordinator, and Candace Fortier, our administration coordinator. And on behalf of the entire team, I'd like to extend a warm welcome to all of you tonight. So tonight's session is entitled Solving the Parkinson's Disease Puzzle. And we all know what Parkinson's disease is, but what we may not realize is that clinicians and scientists are still struggling today to understand why certain people develop Parkinson's disease and others do not. And also, we always ask ourselves is why is it that there's a very specific population of dopaminergic neurons in the brain that are especially vulnerable to neurodegeneration? So there are still so many unanswered questions that baffles researchers today. Tonight, the University of Ottawa Brain and Mind researchers, Dr. Tiago Mestre, Dr. Juan Li, and Dr. Michael Schlossmacher will talk to us about the latest developments in Parkinson's disease research and patient care. And we also have a special guest speaker today who will talk about the lived experience, who we'll introduce later. So it's a real pleasure to begin our session and introduce Dr. Michael Schlossmacher, who is a neurologist at the Ottawa Hospital. He's the director of the neuroscience program at the Ottawa Hospital, the co-director of the Parkinson's Disease Research Consortium, and he also holds the Bhargava Research Chair in Parkinson's disease. He also received numerous awards, including the Grimes Research Career Achievement Award from the Ottawa Hospital for excellence in research and patient care. And he leads a highly productive research team who is committed to developing new treatments for Parkinson's disease. So thank you, Dr. Schlossmacher, for being with us.
Thank you, Ruth, very much for the introduction. Can you hear me? Can everybody hear me very well?
Yes.
Okay, thank you, Ruth. And I hope the video feed works as well. Hello to our audience. Thank you for tuning in and for joining us in this second presentation of the Brain Health Awareness Week. Thanks to our host, Dr. Slack, and her team, and thanks to Mr. Evan Sidel for joining us, and also hello to my colleagues, Dr. Juan Li and Dr. Tiago Mestre. And thank you to the audience, and hello to the audience for joining us under these difficult circumstances with COVID-19 related health scares. So I hope that all of you and your families are doing well. We have a fun program today that tries to dissect 4 different elements of Parkinson's disease research. One is, as you've heard, we'll have a person with lived experience who has been very much involved in advocacy, and we're going to learn from his perspective. We have Dr. Tiago Mestre, who leads a wonderful effort in the Integrated Parkinson's Care Network, what it means in terms of how we can translate some of the research we're doing to improve patient care. And then we have Dr. Juan Li, who is going to talk about prediction of Parkinson's disease. And I have the opportunity to lead off with introducing one topic to you that has garnered a lot of attention, that is the topic of inflammation. So I hope you can all see on the screen here the title that, so the Brain Health Awareness Week 2020, today is on the Solving the Parkinson Puzzle. And the topic that I want to briefly review with you is that some Parkinson's gene insights recently generated information that let us think that some of these genes modulate inflammation. And so that could potentially be a target for future therapy. And I hope you can see that all of you can see the second slide. It's from the Ottawa Tulip Festival, a photo that I took 2 years ago. And that highlights one of the major points that I'm trying to make in these introductory slides, namely that when you look at people with Parkinson's disease, it's very similar to what happens when we look at this beautiful assembly of tulip fields here in the city of Ottawa. Namely that you can see the importance of how genes interact with the environment. All of these tulips that are symbolic for me of people with Parkinson's have pretty much a similar shape. In other words, they meet criteria for Parkinson's, but they have different colors. So there's at least one gene for each of these tulips that different in color that is important to make one tulip be yellow versus red versus purple. But that's only one difference. The other difference is that these are in different flower beds, and that means they have different types of soils. And so there's an environmental factor that interacts with the genetic factor, and to bring out the color, the size, and when they bloom and when they wither away. But so what the take-home message of this slide is as a symbolic representation of in Parkinson's disease, as in many other medical illnesses, there's an interaction between genes that we have and the environment that we're exposed to that generates the outcome, namely what type of tulip and what type of Parkinson's disease we have. And on the next slide, what you see is when we think about precision medicine, which is now very much at the forefront of many different disciplines, think of breast cancer, think of lung cancer, and many other conditions, that we don't lump and group together all patients that suffer from one and presumably the same condition. We actually have learned that in lung cancer, breast cancer, and also in Parkinson's, as much as in dementia, for instance, there's different subtypes. So that in this flower bed, for instance, that you can see here, there's an arrangement according to color. And you see at the very top, a few red tulips, then a larger portion of white tulips, followed by blue tulips, and again in the forefront, kind of maroon colored pink tulips. And I've highlighted on the left some of these forms of Parkinson's that we see in the clinic when we work with patients. There's one at the very top. It's not very frequent, that affects 5 out of 100 people that have early onset Parkinson's disease that starts before age 20, usually, but also can start somewhere between age 15 and 40. So we call these young onset or early onset Parkinson's disease. And then we have one that has a term like GBA1, that's the name of a gene that affects up to 10%. And then in yellow and highlighted in blue here, for the blue tulips, is one gene that I want to tell you about a little bit today. That's called LRRK2. LRRK2 is the acronym for a longer name for the gene. And that affects between 1, but probably 2 out of 100 people with Parkinson's disease that present later in life, so late onset Parkinson's disease. And so on the right, I have highlighted here this summary that from a precision medicine perspective, our goal is to select different subtypes of Parkinson's to enhance the chance that we meet the right treatment for the right disease mechanism for the right intervention in the future. And so that's the motif or the theme behind precision medicine, to match the right person with his or her disease type with his or her treatment in the future. And when one looks at this LRRK2 gene, the variant, so this is the blueprint that we all have. We have DNA, and there's 1 to 2 people out of 100 around the world that have an abnormal sequence for this gene that's called LRRK2. And what's interesting here is there's a number of the building blocks that you see here are the alignment that make this gene a protein. It's a building block that we have in our body and tissues. And you can see at the top of this bar with the colorful arrangement of building blocks, several variants that are linked to disease. And interesting enough, it's not just linked to Parkinson's disease, but it's also to Crohn's disease and leprosy. Crohn's disease is a disease of the gut that causes inflammation. And leprosy is a disease that affects peripheral nerves, and that's caused by a bacterium, Mycobacterium leprae. So interesting enough, in this case where this one gene is not just linked to Parkinson's disease, but variants or abnormal sequence elements are linked to Crohn's disease, leprosy, and Parkinson's. And what we argue all three elements have in common, all three diseases have in common, is abnormal inflammation. And you can see here one of the commonest variant in Parkinson's disease is this one that we call G2019S. So about 1 out of 100 people have this. And so what we thought when we were lucky enough with a colleague of ours, actually several colleagues at 5 different universities in Canada, a total of 9 people, scientists that worked together, and we applied to the government of Canada, received a team award, and we called ourselves CLINT, Canadian LRRK2 Inflammation Team. And what we thought is that LRRK2 gene is representative of how the environment interacts with the genetic makeup that we have. And could it be, because we know, and you'll see in a moment also, that Parkinson's disease, many of these variants are thought to begin in the gut or in the nasal cavity in the nose, that we thought that maybe this is one of the pivotal genes that links the environment with the genetics. And when 2 abnormalities meet, so something like a microbial trigger, like a virus or bacteria, together with the genetic risk, you need both to bring out the disease and not just one or the other. And so we said at that time, let's test whether a virus or bacteria is required to lead to an abnormal outcome in either the lung function or in the spleen or in the gut or in the brain, importantly, if we study this in mice. So we thought, let's study first whether this gene is doing in humans, because a priori, everybody thought that this gene is only doing its job in the brain. But this little bar-coded graph here on the right showed us very early on that the field that has been running for 10 years after this gene's function in the brain is actually not really expressed in the brain. When you look at this green bar here, this is its expression in the bone marrow, and this is its expression in the lung. And the brain is actually this tiny little black symbol here. So here's a gene that everybody thought is linked to knows it's linked to Parkinson's, to Crohn's disease, and to leprosy, but it's actually not highly expressed in the nerve cells or in the brain. So we thought that because it has high expression in the bone marrow where our blood cells, red blood cells, white blood cells are being made, let's test what it does when an animal is being infected. So everything I show you now is just work that we've done in animals, some of which we have corroborated in humans. But in animals, you can control the conditions better. So this is a first experiment where we wanted to bring into a mouse that carries this human abnormality of the LRRK2 gene. And Dr. Bojan Žutinovski in our team he took young mice and infected them with a bacteria, that's Salmonella, that causes in these young mice at a particular dose serious infection. It's called sepsis, and they all die within 10 days normally. And what we looked is whether this gene makes a difference in the survival of these animals. And what you can see here is the survival rate. So these are the normal animals that get infected, and within less than 10 days, essentially every animal has died. And our thought was that maybe this gene, this LRRK2 gene, changes the outcome of this animal's survival. And you can see here's a group of animals that had 1 copy of this abnormal Parkinson's gene, and they lived twice as long. And lo and behold, if they had 2 copies of it, they lived 3 times as long, and which we then subsequently found is happening because this gene increases the degree of fighting this infection and therefore making the animal survive better. But that was a gut disease and a spleen disease and a whole host infection model. So we then took a second model that is mice. This time we use really young mice that get inoculated through the nose because Parkinson's disease often starts in the nose. So we wanted to have a virus, a respiratory enteric virus that causes ultimately, as you'll see in a moment, an infection of the brain. So we wanted to test whether in the brain disease model, not just in the peripheral infection model, this gene makes a difference. And to that, we studied, this is the head of a mouse, entire head, and you can see this area is the brain. Here are the teeth, and this is all the nose. And so we put the virus into this area at the tip of the nose, and it takes a breath, and the virus migrates to the olfactory, so the epithelium where we can smell. And you know that many people with Parkinson's lose their sense of smell. And by the way, this approach, how a virus gets to the brain and affects nerve cells that make us smell, is the same approach how COVID-19 affects many patients that present first with a loss of sense of smell, a loss of taste. So we studied this virus as it enters the nerve cells that mediate sense of smell, and then we can follow it, and the virus then migrates slowly but surely to the brain, into the brain, and causes a disease that's not typical Parkinson's. It's the brain infection, but the cells that Dr. Ruth Slack earlier mentioned, that dopamine cells, are also being lost. So this is an infection of the brain, and we wanted to know whether the Parkinson's disease gene makes a difference. When it helps animals to survive infections better, we were wondering, does it also help animals' bacterial infections? Does it help the virus infection too? And what we saw, surprisingly, is we looked first at mice that are both female and male, a large number of them, and these are the normal mice, and 50% of this chosen dose of the virus die. But we saw a slight difference, and then we looked in more detail at the males, and there's absolutely no difference with the males. But lo and behold, when the female mice were looked at, if they have the normal copy of the gene, they lived, basically 50% of them died by age 21 days after the infection. But here, half of them actually, there's greater than half of the difference. Many more mice that are female with this abnormal copy of the Parkinson's disease gene die. And so that was interesting. So the same mice that live better when they have a bacterial infection, because they mount a stronger immune response, when the immune response happens in the brain, these mice actually were more affected and didn't turn out to have a better survival, but a worse survival. So this is what for us was the model, that if an inflammation reaches the brain, this Parkinson's gene causes a worse outcome. So we then looked at the opposite effect, and that's important because there's at least 5 companies in the world that currently try to inhibit this gene that seems to be overactive. So we looked at a mouse that has an underactive version of this gene. And we again looked in this virus infection model of males and females, and we could again see that the greatest difference when now the animals are protected is in female mice. And then when you have this inactive gene, they live much longer than when they have the overactive gene. And Dr. Juan Li, who you will see in a moment and hear from, she actually then helped us to look at all these many, many hundreds of animals that we looked at. And we looked at it systematically with mathematical means when there's an overactive Parkinson's disease linked variant of this gene versus the protective one. And here you can see that when the females had this infection, had this faulty gene, this Parkinson's gene, their mortality increased significantly. When they were males, it didn't make a difference. And the reason why this is fascinating is because, as you know, in Parkinson's disease, it's usually males that are more affected than females. But in the exception being for this gene, where it's the opposite. In humans, more women that carry this variant have Parkinson's than men. And when we do genetically what the pharmaceutical companies want to do, namely to make this overactive gene underactive, then they had increased survival. So that would suggest that this gene is a good target indeed to inhibit this activity, and we can bring this in future into clinical studies. And the summary of it is that this gene seems to be overactive when it has this version, and this overactivity may help you to fight infections in the periphery, but if the infection reaches the brain, then that makes you have a lesser outcome, a lesser ability to survive as a dopamine cell. And when it's inactivated or when it's underactivated, it seems that in the brain, this inflammation is reduced. So the take-home message from this is simply said that there's several types of Parkinson's, and most types affect men more than women, except for the ones that are associated with a genetic variation in this gene, where women are slightly more frequently affected than men. The typical onset of Parkinson's is probably a combination of 5 different factors, and you will hear about that in a moment, with environment plays a role, the DNA, inflammation, gender, and then the factor of time. We know that the older you get, the higher the risk you have Parkinson's. Some PD-linked genes have a role in immune function, and this could be of relevance not just therapeutically to try to minimize inflammation in our patients, but this is also very important if we target these genes because we may put people at higher risk of infections if we interfere with it too much. And second to last, we may be able to calculate a person's risk to develop Parkinson's, which you will hear in a moment. And then lastly, as you'll then hear from Dr. Tiago Mestre, caring for people with Parkinson's disease becomes more complex. The disease is complex, not just complicated. Many factors come together, and we have to apply a really rigorous multidisciplinary approach, which Dr. Mestre is going to talk to you about in this integrated Parkinson's care network to try to deal with it and to provide the best possible care to our patients and their loved ones. So in my team, I wanted to thank all these people that have contributed to this work. We have collaborated with people at the University of Ottawa that are experts in infectious disease, and also such as Dr. Brown, who's a virologist and who is talking frequently on the air about COVID-19. And you'll hear a little bit about this prediction modeling that we do with Dr. Juan Li and Dr. Tiago Mestre and others. We've been supported generously by different funding agencies. You'll hear a little bit about the funding agencies' tasks in these days. And in particular, also, we're very proud and grateful for the support that we've received from the University of Ottawa Brain and Mind Research Institute. So I'll stop here and thank you. And we'll hand it over to the next speaker.
Thank you, Dr. Schlossmacher, for a great talk. And I'm sure there'll be lots of questions, but if you could hold your questions until the panel discussion at the end, and you can submit your questions in the Q&A box on the bottom of your screen. And so now it's a pleasure to introduce the next speaker, Dr. Juan Li, who is a postdoctoral fellow in the Schlossmacher lab. And she obtained her PhD in engineering in China and later came to Ottawa to develop applied mathematical modeling as a predictive tool for Parkinson's disease. And Dr. Li's research focuses on testing the ability of a mathematical model to differentiate between those with Parkinson's disease versus those without, and how to use this model to predict Parkinson's disease. So thanks very much, Dr. Li. We just have to, okay, here we go.
So it's shows all right?
Yep.
Okay. Hi there. My name is Juan Li, and I'm a statistician and postdoc fellow working in Dr. Michael Schlossmacher's lab. And today I will talk about the PREDIGT score. It is like just Michael briefly introduced, it is a model to distinguish PD patients from the healthy individuals without a movement disorder assessment. So here is a disease trajectory plot for the Parkinson's disease. So as we know, when people age, their risk of developing PD will increase. But for most people, their risk will never high enough for disease onset. But for the idiopathic PD patients, as represented by the solid black curve, and for the young onset PD patients for the red curves, their risk development will be much more rapidly. And there are 3 stages before the disease onset and diagnosis. There are normal stage, preclinical stage, and the prodromal stage. So our goals for the PREDIGT model is for identify at-risk individuals before their disease onset. And the application for this, we have 3 applications. So the first one is for clinical trial recruitment. Represented by this light blue box here, is to identify people at higher risk before their disease onset. And then studies can follow these individuals until their disease development to provide more knowledge about PD. And the second one is for disease prevention and intervention. So for the young onset PD, the risks are more linked to genetic, so it's quite difficult to modify. But for idiopathic PD, there's the interaction between environmental factors, which some can be modified, and also their genetic risk. So by modifying certain risk factors, we hope the disease prevention and intervention can postpone the disease onset, like represented by these two blue arrows here. And the third one is for disease diagnosis. It's just like in our health system, it's often the disease diagnosis starts when your family doctor refer you to see a neurologist. So if you or your family doctor knows better about the risk factors PD, they may notice certain stage, like it indicates you have higher risk or you are like on early stage of PD. So this will enable the patients will get diagnosis and intervention at the earlier possible stage. So here's our hypothesis. So for the PREDIGT score, we think the development of Parkinson's disease is the product of 5 interactive factors. Like the equation here says, so for the E is for environmental exposure, and for D is the DNA and genetic risk. I is the initiation of tissue changes, is the response of the environmental and genetic interaction. And G is for sex, and T is for time and age. And on the right, there's like detailed variables that are included in each factor. So for factor E, we have olfaction, that is actually your sense of smell. So the reduced or lost sense of smell and taste can be a risk factor for Parkinson's disease. And constipation, head trauma, and then exposure to pesticides, including working in the farm, and metal exposure. And here for smoking and caffeine beverage intake, they are actually associated with lower risk. So they have certain degree of protection effect. And then for factor D, we have the genetic risk. But for most people, if we don't really have the detailed genetic information, we can use the family history of PD as a good surrogate. And then for factor I, we have certain non-motor aspects of PD that can also be a risk factor. It's like anxiety, depression, and REM sleep behavior disorders. And also there are 2 biomarkers that are under investigation. That's the alpha-synuclein and tau levels in your spinal fluid. So it has been shown that these 2 levels are lower in the PD patients compared with healthy people. And then we have the sex and age. So here I will use an example to show how we calculate the PREDIGT score. So this slide here is for factor E. Say we have an example you can see here on the blue panel here. So like this one is a male, so he has no exposure to heavy metal and have no exposure to pesticides, and he has no head trauma. Here's the 0 score for these 3 factors. And then he has constipation and increase his risk by 0.5. And he lost his sense of smell and taste. And here is the score 1 indicator higher risk. But he has smoked for less than 10 years and he stopped right now. There is a little bit protection effect. So here we just minus the score by a little bit. And then he drinks coffee regularly over 2 cups a day. And here there's another protection factor and we just reduce the risk a little bit. So in summarize, for factor E, this individual has a score at 1.2 around. So the next slide here is for factor D and factor I. So for factor D, because this individual didn't do any DNA test, so we don't know anything about his details in his genetic risk. But we do know that he has a 2nd-degree relative who has diagnosed with PD. So this will associate with a higher risk of developing PD by 0.25. And then for the score I, is that this individual doesn't have any depression, anxiety, or REM sleep behavior disorder. Here's all the 0. But by doing the lumbar puncture, we find that his alpha-synuclein level and tau level are lower than like the threshold. So it's indicated with higher risk. So here is like 2 scores associated with higher risk. And then this slide is like, we just add the scores for factor E and D and I. We have this summary score. And being a male, there's like a higher risk. So we times 1.2 and times his age at 73 when we do the study. So we have a total score with 169.7. So this is only an example for one individual, how we calculate the PREDIGT score. But to see how the model perform, we have to put it in a real data set and compare with other individuals to get more like statistical like results. So here we have 2 data sets. It's called DeNoPa and PPMI. So DeNoPa is the de novo Parkinson study. That's a Germany study and is only in 1 center. And we have PD patients, 159 PD patients and 110 healthy controls. And then PPMI is a North American-based multi-center study. And we have 430 PD patients and around 200 healthy controls. So note here, we have the age and sex matched controls. That means for the controls, those 2 studies, they control the ratio of the sex ratio and the age distribution as represented here. So we first look at our model's performance in the DeNoPa study. The first step is for each variables here, we want to see what's the risk effect size represented in this data set. So here's the odds ratio. So like the odds ratio, so for odds ratio higher than 1, that means associated with higher risk, and below than 1, that means is associated with lower risk, like a protection factor. So here, this right vertical lines is for 1, that's the neutral fact. And then for this dot here is for odds ratio, and here is for the confidence interval. So the higher the value here means high, like associated with, like more associated with higher risk. So here we can see for factor E, olfaction, reduction, and constipation are really significant and associated with higher risk. And smoking and caffeine shows some protection factors, but for other variables, they're not like statistically significant. And then we have family history of PD, but note for this data set, we don't have their genetic information. And also for factor I, depression, anxiety, and RBD there, and also for these 2 biomarkers, they all like significantly associated with higher risk. And for sex and age, they are actually like really informative risk factors with PD, but because these cohorts are sex and age matched, it doesn't really show here, but like in the population, we will consider age and gender as a risk factor. So here are the model performance. This is like calculating the score for each individual in this cohort. And so here, this plot A and plot B, they actually convey the same information. So you can choose which plot you feel more comfortable and natural to you. And they all say the same thing, is that for light blue is for the healthy individuals, and for pink is for the PD patients. And for most healthy people, they will have a lower PREDIGT score compared with the PD patients. That's basically what this 2 plots shows. And so, but the model will confuse a little bit for this overlap region. That's why it can't like perfectly separate these 2 cohorts. These 2 groups. And here AUC is a measurement of how this model performed, and it's quite technical and may like beyond the scope of this talk. But a rule of thumb is the value of AUC is generally between 0.5 to 1, and which 0.5 means it's just a random guess, like you toss a coin and say if you have PD or not. And for 1, it's like perfectly separates these two groups. So here our model's performance is 0.85, is actually quite accurate. And to be more specific, if we use the 78.2 as the threshold to differentiate PD patients from healthy control, the sensitivity is like we can correctly identify 69% of PD patients, and we can correctly identify 88% of the healthy people. The next slide is for PPMI. That's another cohort is in North America, and we have a larger sample size for this cohort. And like for this cohort, certain wearables were not captured. So we can only work with the wearables that are already collected in this cohort, but it shows a similar effect as in PPMI, like olfaction, constipation, family history, genetic, anxiety, depression, RBD, and biomarkers, they all significantly associate with higher risk. And same problem as DeNoPa, like sex and age were not correctly represented. But they are confirm our hypothesis, like the selected wearables are associated with higher risk of PD. And similar to the earlier slides, here is the model performance for the PPMI. Same again, we can say for the blue distribution, that's for the healthy control, their scores are generally lower than for the PD patients. And actually this model does better than in DeNoPa. Is like the total performance AUC is 0.88, and it can correctly identify 81% of PD patients and 81% of the healthy people. So the last result here is like we try to combine those two cohorts, and because like these two cohorts may collect different information, so we use like fewer wearables in that model, but still we can see here the score difference is still there, like the PD patients have lower score compared with the healthy control. And more important here for this plot, the AUC I mentioned before is actually is the area under this curve. So this curve called ROC curve. And we can see here for this model, it's actually have really similar AUC and ROC curves for these 3 data sets. That means our model is really robust. So it's not biased for one population. It's actually their performance very balanced and robust across all the population. So it gives us confidence that we'll test it later in the larger population and may later like use it in a clinical settings. So here for next steps, what we already done report here is for aim 1.1 is the discriminative power of this model is to how well it can differentiate PD patients from healthy controls. And then for predict power, like we need a new data set that is a prospective data set. That is, it identify a group of people with certain traits or risk factors of PD and follow them for a certain period of time, or often several years, until disease onset. And those data sets will be used for us to test the predictive power of this model. And optimization is here because our model was hypothesis driven. So all the variables and their assigned values are like quite conceptual, and then use a large data sets. We'll just fine tuning all those values and make this model performs even better. And then calibration is that because earlier we only use case control cohorts and it's not in the general population. For example, like in those cohorts, we have maybe 2/3 are PD patients and only 1/3 are healthy control. While in the population settings, the majority will be will be people don't have PD. So for calibration, it's like we put this model into a population setting and see how this model performed and change all those variables based on the incidence in the population. And the third aim is the tool development. Is like we want to translate our model into an easy to use questionnaire, or it's the kind of a questionnaire like you can use like in your doctor's office. There's certain like maybe 10 to 12 questions, and you answer yes or no or other responses, and then we'll calculate your risk of PD. And this score can be used for, say, doctor referral, or just if you are interested in your general risk. So the ultimate goal here is to predict the risk of developing PD in still healthy individuals before their disease onset. So at last, I want to just thank my colleagues in the Dr. Schwarzmacher's lab and our collaborators, and also more importantly for the patients and subjects who share their data in this cohort. And I also want to thank our funding agencies. That's all. Thank you.
Thank you very much, Dr. Li, for a great talk. And I'm sure we'll have questions, so please submit your questions in the Q&A box down below, and we can discuss those later on at the end of the session. So now I'd like to introduce our next speaker, Dr. Tiago Mestre, who is a neurologist at the Ottawa Hospital. He received an Ontario Early Researcher Award for his research excellence, and he has co-led and developed the Integrated Parkinson's Care Network, which is an international network designed to transform patient care. And this is a huge accomplishment for any investigator, let alone an early career researcher. So thank you, Dr. Mestre, for being with us today.
Thank you, Ruth, for this kind presentation. So I'm just going to share my screen. Okay. So following the title of today's presentation, I decided to call this specific Solving the Puzzle of PD Care. So we talked about molecular biology, mechanisms of disease, how to predict and differentiate between people with Parkinson's disease versus healthy controls. In this talk, I'll try to talk about the efforts to optimize the care delivery to all people living with Parkinson's disease. And so, and why? So, what are the different factors that make it challenge to deliver care in Parkinson's disease? So the puzzle of Parkinson's disease care. So as many of you would know, Parkinson's disease is a chronic condition in which people accrue multiple symptoms throughout their lives and develop increasing disability. These symptoms are not only motor, but they are non-motor. And you see in this slide different aspects of the disease which make it particularly challenging. And I say that as a neurologist, we were not, usually it's not part of a training of neurologists to take care, for example, of gastrointestinal symptoms, or even to some extent autonomic symptoms. I think that in mind, we also know that the incidence of Parkinson's disease, one of the main predictors, as Dr. Juan Li demonstrated, is age, and has especially the Western population, but I would say in other areas of the world, as the general population ages, also the incidence of Parkinson's disease is expected to increase, and it can double by 2030. Also, there's the systemic aspect, the societal aspect of how societies can organize themselves to deliver care, not only to, I would say, to Parkinson's disease, but other chronic disabling disorders. Definitely the idea that multiple healthcare professionals and care resources can talk to each other to deliver care to these people and in Parkinson's disease, it's very attractive. But as we started to review the literature, we found that the models that were tested, and one was particularly common of having a multidisciplinary clinic, so all the resources in a given physical location, or having a network, in this case, a national network that was developed in the Netherlands by my colleague Bas Blom, these studies, by whatever reason, they've not showed a robust benefit. And also the aspects of economic sustainability came to the surface for these both models. So here in Ottawa, together with my colleague, Dr. Grimes, that has co-led this effort, we tried to come up, and already in 2014, so 6 years have passed, can we deliver care in a pragmatic and efficient way? So this idea of a one-stop shop for Parkinson's disease, can we include technology to improve communications with our patients? And how can we do it if ultimately we have 24 hours in a day and 7 days in a week? And so can we, and these are some ideas that populated our minds, can we use more efficiently already the resources we have in our community and in the hospitals? And of course, we're thinking primarily in that initial stage in Ottawa. And can we break silos in our health system to achieve what we call integration of care? And ultimately, can we deliver care that, that is focused on the patient's needs? And so came this idea of the integrated Parkinson's care network, which I call the Ottawa model. And I will call your attention to this diagram. So in the IPCN, we're trying to have this patient-centered approach. One of the first initial aspects is to, as we developed this idea, we brought the input from care partners, from people with Parkinson's disease. We also brought this idea of care integration that it's explained in more detail in the left side of the slide. So this idea of tailoring care plans to individual patients, selecting what is what we call the care priority that we need to focus on and hopefully have the greatest impact in the lives of people with Parkinson's. Also, create this idea of a virtual care network that basically tries to potentiate the healthcare resources that are already available and optimize the access and the use of those resources. And then to promote this patient-centeredness, try to increase patient empowerment through knowledge and skills acquisition in terms of managing life with Parkinson's disease, but also in a more broader way of living healthy. Okay, so this idea of self-management of knowledge, skills, and confidence to manage necessary daily tasks and to live well with Parkinson's disease. We also had planned involving technology, including technology, to enable all these concepts, and something I will talk later during my presentation. I'd like to highlight the role of Diane Coté. So she's a Parkinson's disease nurse at our center, and she has been a key driver of the development of this model as what we call a clinical care integrator. The next few slides are going to talk about the first formal study that we completed in 2018/19. We did it with funding available from the PSI Foundation, but also the Parkinson's Research Consortium and the University of Ottawa Brain and Mind Research Institute. And this was a very pilot study. So we looked at what was the experience of people with Parkinson's disease undergoing this 6-month program of the IPCN. We looked both at patients that were recently diagnosed, but also at people that had Parkinson's for longer and intuitively would have more complex care and more care needs. And so we wanted to know what was the experience, but also if we could deliver the IPCN as envisioned. And again, I would like to emphasize that it was a multidisciplinary research team effort and whole based in Ottawa, involving the Ottawa Hospital, the Ottawa Hospital Research Institute, the Bruyère Institute, and also the active role of the advisory board, both for patients and care partners. Briefly, what did we show in terms of can we deliver the IPCN as envisioned? So I was quite happy that we were able to recruit 100 participants in 6 months. usually, and has a comparisons, a clinical trial testing a new treatment for Parkinson's, we would recruit in about the same time, perhaps 5 to 10 people. So overall, the recruitment was quite encouraging. We did find that for a significant portion of participants that were eligible to take part in the trial, but did not make it, some aspects that require improvement on our side and are currently being addressed, we need to list them. So traveling distance, access to transportation or language barriers. So these are information that is very important for us so that we can improve this care delivery model. And then we also looked at what are the care priorities overall, and that's more a measure of the people with more advanced Parkinson's disease, but also in the newly diagnosed patients. And you see things such as speech and communication, anxiety, depression, mobility, balance, fall. So these are common aspects that the network, the IPCN, needs to be able to deliver proper care. And by defining priorities, we can better tailor our development of the network and also allocate more resources to these areas that perhaps are more required and requested. But then if you look at the newly diagnosed patients, the aspects of coping strategies, and I would say learning to live now in the future with Parkinson's disease was quite relevant. And then we tried to have preliminary data on if the IPCN program was actually impacting the lives of people with Parkinson's disease. And what we found is that the perception of care improved overall in the entire study for the whole, in general, for all participants. And this is what listed here as the PASIC plus scale. If you look at quality of life, the IPCN seemed to impact to a greater extent in the people with more advanced Parkinson's disease. But overall, the aspect of satisfaction, I think, is also very important, was found in more than 95% of the cases, both for patient care partner. We also look at cost. So ideally, we look at this intervention as being deployable on a systemic basis to, for example, the Ontario healthcare system. And so having cost associated with benefit is very important. And again, in terms of initial data, these are encouraging data. And we did what's called a return of investment analysis that seems to suggest that for each dollar invested in the program, both developing and maintaining it, there's a potential return of almost $5 in terms of reduction of societal costs. And so what is next? Some aspects Dr. Rutzak has already mentioned, but let's start with the technology. So our vision is that many of the care concepts that we want to enable with the IPCN, such as the self-management, the patient empowerment, can be delivered potentially by a patient portal that can be used by patients and people with Parkinson's disease and care partners. And we started this development of this vision both with the Ottawa Valley-based company Macadamian, but also with another collaboration in the University of Ottawa with the School of Communication, Professor Sylvie Grosjean and Professor Luc Bonneville. And as previously done, we started by asking people with Parkinson's disease and caregivers, how could technology impact their lives? And this generated the first prototype that actually now is being tested by 30 participants as we speak, that we named the eCare PD. In a nutshell, what is the user experience? So they have a welcoming tutorial that teaches them how to use the platform, a home screen with a calendar where they can schedule their activities related with Parkinson's disease or other activities. And then they go on defining their care priorities that then they can track through a questionnaire, potentially in the future with data collected in wearable devices or other quantification methods. And also, finally, we have more general education resources that any person using this platform can enjoy and integrate in their lives. Also, an important project is the opportunity of making the IPCN international model, and we've done so by collaboration with 7 other European institutions in a grant funded by CHR, which is a governmental funding agency in Canada, but also a European grading agency called JPND. And I'm honored to say that the University of Ottawa is leading this project. It's called iCarePD. You can look at more details both in the Twitter account and more extensive information in our site, icarepd.ca. And there's multiple projects that we are coordinating. The overall idea is, how can you translate these concepts of care that I've explained in different cultural and different cultural and social environments and different healthcare systems. So we're talking about Canada, Spain, Portugal, Italy, France, Germany, Czech Republic, and Ireland. And so we're looking of aspects of co-design, so bringing patient input in all these different contexts to make a better IPCN, but also economical aspects, but also in terms of health geography, which is led also by Professor Eric Crichton based in our university, looking of how can we access resources and what are the changes that need to be implemented for a better access to those resources based only on their geographical location, but also developing a more ambitious and more complex e-care PD, and finally testing it as we've done in Ottawa, but testing in all these different centers. We're also looking at the ethical and legal aspects of self-management, use of technology, that also is led by Professor Jennifer Chandler, that's also based in University of Ottawa. And this is, I hope that in a future session, I'm able to bring more the results of this development of this international network, because ultimately what I would desire and envision is that we are able to bring in a pragmatic and sustainable fashion, improve care for people with Parkinson's disease. And this is my last slide. I'd like to thank all in the Movement Disorders Group that have enable this project and other research projects. I think ultimately impact the solving of the solution of the Parkinson's disease puzzle. Thank you for listening.
Thank you, Dr. Mestre. And I think Dr. Mestre, you will introduce the next speaker.
Yeah, so it's I think I'll stop sharing. So it's a pleasure to present the next speaker, Mr. Haven Siedel. So I kind of debated which aspects of his life should I showcase for today. I would like to say that I've come to know Mr. Haven Siedel since 2014, where I first met him when he took part in one of our trials in Parkinson's disease. He had been diagnosed with Parkinson's disease. I think since then he has given examples how can we enable ourselves after being given a diagnosis and make of that a cause. And so in this time, Mr. Haven Siedel have advocated for Parkinson's disease, creating a fundraising event where he puts together the spirit of advocacy, but also his love for cycling, and is also part of the board of directors of the Davis Finney Foundation based in the US, and actually was able to bring one of their meetings to Ottawa. And without further ado, I'll leave the stage for Haven. Thank you.
Thank you, Dr. Mestre. I guess you can't say for ethical reasons that Dr. Mestre is my movement disorder neurologist, and I've been in his care for many years, and I'm delighted to help out today and talk a little bit about my own experience. I'll start with the fact that you probably all know, and that's that this is a condition that is no fun. And many of you will work in your own areas. You may work on a cure, you may work on titration of medicine, you may work on other treatments, modalities, etc. And I would just want to explain to you 4 aspects of the experience of having Parkinson's. The first is it's obviously a complex condition, and it differs among so many of us. You know, our clinicians will know that we have often take medication. We'll deal with a physiotherapist. We may deal with a speech therapist, a pharmacist. Never thought I'd be so close to my pharmacist. Acupuncture, chiropractic, massage, spiritual consultants, mental health physicians as well, and caregivers. And then, you know, people will throw all kinds of quack ideas at you. I know enough to know that double-blind studies are important, and the placebo effect is awfully powerful with this condition. But what it means is you're actually quite confused by the complexity of it. And the lived experience of that is, it's hard to explain, and it's hard really to know unless you live it. That comes with a certain level of ambiguity and uncertainty about the progress of one's condition that can be stifling. And it can make one a little fearful. I'll tell you that my best treatment is denial. You know, if you start thinking about your future, it can become quite overwhelming. And I'll say a little bit more about that in a second. The social aspects, of course, there's psychological aspects of the disease, many different conditions which Dr. Mestre referred to. But people can be shut in because they're so self-conscious. During this time of COVID, it's particularly difficult for some of those folks, especially with advanced conditions. You can be overwhelmed with fears of the future. Some of us experience pain and cognitive compromise as well. So this idea of fear and helplessness is something that really can be something that's hard to appreciate, and I think we all experience from time to time. And it's important that people not feel victimized, I think, by that. And I've had great success in my life. I'm CEO of an organization. People look at me as somebody who's achieved a lot. And that comes with it. I suppose it came with a sense of duty, but I also resolved, after talking to Davis Finney, that, you know, the only thing I could do, I can't work on a cure. I can't learn these different treatment modalities. I have to be exposed to those by practitioners. And the only thing I could do is try to live well today. And Davis came to that realization. He was himself a cyclist, that somebody said, don't worry, it'll be a cure in 5 years. 5 years passed, and after 10 years, somebody said there'll be a cure after 10 years. And of course that passed. And he resolved to help people live well today. And I was quite inspired by that because it was something that I could do. And it was that same spirit that led me to Dr. Mestre and the study that I participated in, but led me to co-found the Growling Beaver, a fundraiser for Parkinson, now Parkinson Canada, in addition to the Davis Finney Foundation, that has now, after 5 years and 6 events, raised just shy of $2 million. This year, we've raised $211,000 in a virtual event, the largest single fundraiser for Parkinson Canada. I'm quite proud of that. Not personally, but because of the community we've attracted around it. And there's some magic about this event and the idea of living well today that has really inspired some people. For me, and I'll leave you with this thought, which is a hopeful thought. And that is that, you know, one would look at me and think that I'm a pretty happy person given what I've achieved. But I was quite transformed by the experience of the Growling Beaver and the joy that gives to me. And if you don't know this little book by David Brooks, somebody gave it to me a year ago. I've come to understand that Parkinson's is actually my chance for joy. You'll remember that Michael Fox wrote a book called Lucky Man about his own experience. And he said, and he was living a rather hedonistic life, that if God or Yahweh or Allah or Mother Nature, whomever you believe in, came into his room and said, I could restore you to the person you were before you had Parkinson's, he would tell them to go away. And I think it's because he's experienced something like this. And Brooks writes about the difference between happiness and joy. And he says that often we say a good life is a happy life. We live, as it says in our founding document, the pursuit of happiness, as we say in the US founding document, the pursuit of happiness. The difference between happiness and joy is that happiness involves a victory for the self, an expansion of the self. It comes as we move toward our goals, when things go our way. Joy tends to involve some transcendence of the self, some doing something for others. And while we can help create happiness, we are seized by joy. That while happiness is good, joy is better. And I found that for me, I'm better for having Parkinson's, and it helps me deal with the day-to-day. And that kind of inspiration lightens my load, and I hope it helps other people. And I will conclude there.
Thank you very much, Mr. Evans-Siddell. That was a very inspiring talk. Really appreciate it.
Amazing. Yeah, thank you.
And thank you to all the speakers. And I think at this point, we can open the session up for discussion. And again, questions in our Q&A box. And I'll ask Natasha Hollywood, our program manager, to lead the panel discussion.
Thank you, Ruth. So I'll have the speakers, the panelists, maybe to open up their cameras, and we'll start with a few questions. The first question is to the panel, actually. So what we think would be the person to answer would be, so deep brain stimulation works quite effectively in some forms of epilepsy. Has it been tried extensively in Parkinson's disease? If it has been, what were the outcomes?
I can perhaps address that question. Another hat I carry in Ottawa. I'm the co-director of the Deep Brain Stimulation Program for movement disorders. So actually, it has been an established therapy now for about 30 years, and so it can be quite impactful in people with Parkinson's disease, especially at a stage of the Parkinson's when the medications don't last as long as they supposed, or they comes with involuntary movements that we call dyskinesias, or for people that have very severe tremors that do not respond to medications, and that can also happen. And so it's definitely an established therapy for Parkinson's disease.
Thank you, Dr. Mestre. This question is for Dr. Schalschmacher. Since the incidence of PD is many times caused by activation of LRRK2 gene due to bacterial or viral infection, are the epigenetic effects present in some major way?
What effects present in a major way? Sorry.
Epigenetic
Okay.
effects, yeah.
So epigenetic means that your DNA is changed by various events that happen to you in life. So your mother and your father gives you a copy of their blueprint, and that's called DNA. And then through various events, your DNA can actually be further modified or edited. And that's by changing what genes can be actually turned into products and which genes are essentially turned off. So that's a very good question. And that field has just really started to take off in the last few years. It was in part driven by a Canadian woman named Vivienne Lefebvre. She tragically died at the age of 42 just 2 months ago when she was hit by a truck. But the notion that what happens to us when we go through infections, whether it's a virus infection or a bacterium infection, and how that changes how our body responds is one of the most exciting areas of research currently, to try to see whether that accelerates Parkinson's disease development and increases the risk, or whether it decreases it. And this LRRK2 gene seems to play a role in it. And whether the LRRK2 gene is affected by epigenetic change or not is not yet known. what's interesting is that there's forms of this gene that actually protect you from Parkinson's. This was an outcome of very large international genetic studies where 500,000 people with Parkinson's were included all around the world and compared to controls. And so people found that, yes, it seems to be often in up to 2% of cases involved in developing Parkinson's, but then they also found that certain variants can protect against Parkinson's. And so this is, in short answers, this is a hotly explored area, and we probably will know whether epigenetic effects play a significant role in that gene in the next 5 to 7 years, I hope.
Dr. Li, this is for you. How will the model that you presented, how will this model affect our life? And when will the questionnaire be ready to use?
Yeah, thank you. So for as I talk, like I talked in my presentation, so we have like 3 applications for the model. It's for recruitment for the clinical trial and for disease prevention intervention, and also for like a disease diagnosis. So I think they all affect us in the Parkinson field. And also as for the model, as for the tool and the questionnaires that implements this model, we are working on, we are just like working on a pilot project, giving if we have the enough funding, and then we'll pilot it in Ottawa, and we'll have like a small study that we have a prototype of the questionnaire based on our model, and we will like provide those questionnaires to our patients or family members if they're interested, and then we can see how this questionnaire performed in a general, like it's not so controlled environment, it's more like a population setting.
Thank you, Dr. Li. The next question, it says relationship of Parkinson's disease and dementia. So it could be, is there a relationship with Parkinson's disease and dementia, or explain maybe?
Should I take that one? Yeah. Okay, so that's a very good question. We know that this also is linked to what type of Parkinson's one has with the detailed cellular, biochemical, genetic, and molecular processes that are going on. In about 7 out of 10 people with Parkinson's disease who have the typical changes or shared changes under the microscope, if one were to do an analysis of the brain, one can find changes that progress and definitely spread beyond the movement control centers in the brain. So not just where the dopamine cells live, but also other areas. And Dr. Mestre mentioned earlier, many patients have autonomic changes that affect the peripheral nervous system. And we know that according to one particular hypothesis, with progression in age, other areas in the forebrain are affected that control thinking, sharpness of intellect, judgment, reasoning, and memory. So as many as 7 out of 10 people that have this late onset Parkinson's disease, they're at risk of having cognitive problems. What's interesting is on the flip side, people who have very early onset Parkinson's disease that have, for many reasons, as I tried to show with the tulips, a slightly different form of Parkinson's, they seem to, although their disease starts so early, their motor illness starts so early, they seem to have less of a risk, a smaller risk of, or no risk, depending on what type of genetic defect they have to develop cognitive problems later on. Having said that, all of the above is complicated by what we heard from Mr. Siddall earlier too, that, the effects of pharmacotherapy, he's now close to his pharmacist because he has to take medicines on a regular basis. And medicines that we give our patients to help them with movement problems can also affect thinking, can affect cognition. And when people have depression, there's this entity that's called depression causing pseudo-dementia, so that people that are actually depressed and not effectively treated, they can look like they have a cognitive problem. So all to say is that this entire IPCN network, this integrated Parkinson's care network, this approach to the disease frequently and invariably takes into account the cognitive health, the emotional health, so psychiatric issues, and to make sure that people are being assessed for it, whether they have depression and or whether they have cognitive changes. So that's all part of this complexity that Mr. Siddal also described so eloquently, and saying that this is a very complex disease entity.
Thank you. Good evening. I saw that exposure to metals is one of the risk factors of Parkinson's. Can someone elaborate on that?
Juan, do you want to comment on that, how it showed up in your studies?
Yeah, okay. So well, this is more based on our data dictionary from the DeNoPa. Just for PPMI, I don't really have like more information on that. But for DeNoPa, they ask certain questions. But here, one thing is for metal exposure, it's more like a long-term community phase, like job related. So actually, the real term or like more accurate question is like for your work, have you follow like have you ever worked in following area like welding or metal smelting or galvanizing? It's like some metal related work, so it's a long-term effect. And also if you have like heavy metal poisoning, so I think it's more serious and like cumulative effect. So it's actually for this data sets and for the population, like the incidence rate of this risk factor is actually really low, and it doesn't have a really strong effect size. But it's like really to complete our model because there's some evidence that is associated with the higher risk of PD.
Yeah, and I would just add that I think that's a proper and really good answer. What has come out of the drug user community related epidemiological research too, there was a famous paper that was published about 10-15 years ago, that a number of drug addicts in Lithuania and in Latvia, in the Baltic areas, had cooked a new drug that they were injecting themselves, and it was tragically contaminated by very high levels of manganese. So among divalent ions and metals, manganese seems to be a particularly toxic component for the areas in the brain that are affected by Parkinson's, followed by others like cadmium. But manganese seems to be also, from a welding perspective, involved in elevating your risk.
And perhaps I would just add that, as Dr. Schulzmacher was highlighting, not necessarily anyone that looks like having Parkinson's disease will have Parkinson's disease. So there's different factors that can cause the neuronal death in the areas that are related to Parkinson's disease.
Okay, for timing, I'll ask maybe 2 more questions. Dr. Mestre, this one's for you. Are you still recruiting patients in the iCare PD program?
So we, at this point, the iCare PD program and the IPCN program are still research. So we'll be recruiting participants as we do new studies. We currently will have one ongoing the next couple of months, and likely one for the international consortium next year, providing that COVID allow us to. But ultimately, I think the idea is that for any patient, at least in our center in the future, and I think hopefully in Ontario, I would say across Canada, would be able to provide the core care concepts of the IPCN for any patient entering the clinic. And that's our main ambition, ultimately, that we are able to include in the budget of the Ministry of Health, provincially and federally, a cost that allows for this to function. And we want it to be very pragmatic and feasible. So that's our ultimate vision.
Okay, great. And one question. Exercise is obviously important, but there seems to be little guidance regarding level of effort for PD patients. How aerobic should one be on a regular basis? So how aerobic should one be on a regular basis? Maybe Evan or others can answer that.
I can share my experience. I can't give good advice on the medical aspects of it. I was given advice early on by Davis Finney to exercise frequently, but not to the point of fatigue. And my own experience is that when I'm particularly intense with exercise, my symptoms present themselves. It's almost like my dopamine levels are just dissipated. And then I'll get very neurological, I'll have active tremors. So that's my experience.
I would add to that, I think that's a very good observation. I think this also has something to do with excessive amount of exercise or very strenuous efforts. You increase your adrenaline level to make the heart pump more efficiently and faster. And adrenaline, we know, is just a multiplier of tremor activity. And so I think that the advice that you received was really good. And it turns out that most patients experience what works for them really well, and also what type of recovery exercises like cooling down and compensatory or complementary exercises. I think that there's not one magic exercise for people. It depends on age, sex, preferences, and past history of exercises. So I think the important thing is to actually try out multiple different forms of exercises.
Thank you. And Evan, just so you know, there were a few statements of appreciation for your talk. They were very appreciative. And one last question for you, if you'd like. Does stress increase the severity of the symptoms? Good question.
Yeah, I think it does. Lack of sleep seems to, alcohol seems to, in my case. Dr. Mestre knows all this stuff, but stress does for sure. And unfortunately, I tend to relieve my anxiety by pouring myself into work. It's a bad circle. So yeah, that's my experience. I don't know if that's clinically demonstrated, but.
Well, thank you. Thank you all the speakers and panelists. Thanks to everyone for their questions. I'll pass it back to Dr. Slack, and I greatly appreciate all the feedback here. That's great. Thank you so much.
All right. And I'd also like to thank all the speakers for great presentations. I'd also like to thank all of our partners. And of course, today we're highlighting our partnership and collaboration with the Ottawa Hospital, the Parkinson's Research Consortium for very productive collaborations. I'd also like to thank the Brain and Mind team and all of you for being here tonight. And one thing the pandemic has taught us is that medical research is critically important. And so if you'd like to support research at the University of Ottawa Brain and Mind Research Institute, you can go to the link on our website and donate. And if you would just like to get more information on Parkinson's disease or attend our future events, that's also on our website, or follow us on Facebook and Twitter. So I hope you can join us for tomorrow's event. And it's about your brain and exercise and why it matters. And that also applies to Parkinson's disease. So thank you everyone for being with us today. Good night.